So, as far as the catabolism of amino acid is concerned, one of the very important part of the amino acid is the amino group. So, first you have to get rid of the amino group, and then other part of the amino acid can be degraded. One way of doing this is a process in which the amino group of many amino acids is transferred to a keto group, making a common amino acid. For instance, in the case of let us say one, we say that there is an alpha amino acid, S can be any amino acid, and it gives this amino group to a keto acid, alpha ketoglutarate, and you get glutamate, and you get a keto acid also. So, in this way you can see the amino group which is received by the ketoglutarate. In that way, the glutarate acts as the acceptor of the amino group, while the amino acid, of course, the donor of the amino group. You get the glutamate, and of course, along with this you get a keto acid. So, there are lots of amino acids in which transamination occurs, and you get a common amino acid as a result of that transamination, that is the glutamate. Now, also keep in mind that it is a very specific reaction. I mean, aminotransferases are of course the enzymes which are involved in this to carry out this transfer of amino group. So, this is a quite specific for every amino acid. There has to be a specific aminotransferase. There are two very important, very common aminotransferases are alanine transferases and the aspartate transferases. So, you can see here that alanine transferase gives this amine group to alpha ketoglutarate, as we have just seen, and you get glutamate, and you get pyruvate as a keto acid. And then you also get as far as for the aspartate, you get you from aspartate you give the amino acid to alpha ketoglutarate, and you get again the glutamate, while the keto acid is the oxaloacetate. So, in this way you can see that as a result of this transamination, I mean, of course, you are not getting rid of the amino group. It is only moving from one amino acid to other to make another amino acid. But the thing is that you get a common amino acid, glutamate, which can be then acted upon by deaminase, which will lead to the deamination. So, that is the reason that is the benefit of transamination. Also, one other aspect of transamination you should keep in mind that pyridoxal phosphate PLP, it is a coenzyme which help in this transamination process, and we have been seeing it in all sorts of different metabolic pathways which we have been studying, that these coenzymes, these cofactors, coenzymes, or any other such compounds which do not become the part of the product, which are neither reactant nor products, but still they play very important, actually indispensable role in the formation of the product and carrying out the reaction successfully. So, here whether from a formation of from aspartate, whether transamination of aspartate is concerned, or whether transamination of alanine is concerned, or any other amino acid is concerned, the PLP plays a very important role in it. Of course, then as far as this once the what happens actually is that the PLP gets the amino group, and that amino group is then given to a keto acid, and you get the glutamate at the end, and as a result of this, the original PLP is retained or regenerated. So, what happens that shortly amino acid gives its amino acid to a keto acid to form the glutamate by giving the amino acid first to the PLP. PLP then donate this amino acid to a keto acid to make the glutamate, and the original PLP, that is PLP without an amino group, is regenerated. Of course, as I have told you, amino acids are such an important part of your metabolic pathways. So, any defects in their metabolism have some medical implications also. Sometimes you see that your you see in your blood there is much more aminotransferase uh, levels as compared to normal levels, and that is an indication that there is some problem with your liver, because liver sometimes it can be inflamed, it can be affected by infection, or for whatever other sort of damages. It will lead to the leakage of these transferases, this enzyme, into the blood plasma, and as a result this act as a very important diagnostic characteristic, diagnostic test to find out whether or not your liver is functioning well. If liver is not functioning well, you will get more, I mean, if there is more aminotransferases than the normal level, it means that the liver has been damaged, or there is some problem with the liver, whether it's because of some infection like hepatitis, or like from any other shock to the liver. Not only liver, but also for not only for I mean, there are two important, let me tell you also, there are two important transferases I have already told you. One is the alanine transferase, and then other is aspartate transferase. Alanine transferase is used to as a diagnostic marker. Its level is used as a diagnostic marker to see whether or not a liver is functioning well. While aspartate is more general, it's not, it's very sensitive, but it's not specific for the liver. So if there is more aspartate level, it means that other than liver, maybe muscular, or maybe there are some problem with your heart. So as I have told you, the elevated level of aminotransferases can tell you, as a diagnostic marker, can tell you that what sort of damage has occurred. It may be liver in case of ALT levels are high, or it can be any other disease other than the liver, if not ALT but the AST levels are high. So also you can see here that if your liver is not functioning well, one of the another marker is the bilirubin. Its level also become very high, and you can see here that as soon as the ALT levels gets very high, you can also see a very sharp rise in the level of bilirubin, which shows that something really bad has happened with the liver.